and I will set a playlist of signal. I will talk about the signal analysis and the receptors. So we will start with the first lecture. The first question is, what is the function of acetylcholine esterase? So acetylcholine esterase, because it ends with the esterase, so you know now it's an enzyme that plays a crucial role in the nervous system by breaking down the neurotransmitter acetylcholine. It's primarily found at the neuromuscular junction and synapses of cholinergic neurons, where it serves the following functions. And then we discuss what are the functions of acetylcholine esterase. Actually, we want to know what is acetylcholine. Acetylcholine is a neurotransmitter. It's just maybe founded in a neuromuscular junction that's made between the neuron and the muscle, or synapses between the neuron and the neuron. And the acetylcholine, it causes the muscular contractions and other functions. The acetylcholine esterase just to break down the acetylcholine. This is the main function of acetylcholine esterase. So, acetylcholine esterase is an enzyme that plays a crucial role in the nervous system by breaking down the neurotransmitter acetylcholine. It's primarily found at the neuromuscular junctions and synapses of cholinergic neurons, where it serves the following functions. So, the first function, termination of nerve signal transmission. How does this happen? Acetylcholine is released from the presynaptic neuron into the synaptic cleft to bind with the receptors on the postsynaptic cell, like a muscle or a neuron, triggering a response like a muscle contraction or a neural signaling. We want just to know what is happened here. By breaking down the acetylcholine, it's just making termination of the nerve signal transmission. So the first part, I just explained what is the functions of acetylcholine. So we have a presynaptic neuron, postsynaptic neuron, or a cell like a muscle or a neuron, and in the middle there's something is called synaptic cleft. So the acetylcholine is released from the presynaptic neuron into synaptic cleft, and then bind to the receptor that is present in the postsynaptic cells, whatever it's a neuron or muscle, and that is lead to the neuron signaling of the postsynaptic cell is a neuron, or muscular contraction if the postsynaptic cell is a muscle. I'm talking about the acetylcholine. Then it happened, the acetylcholine esterase, which is the enzyme that we are talking about, rapidly breaks down the acetylcholine into its inactive metabolite, which is known as acetate and choline, after the neurotransmitter has bound to its receptor and initiated the signal. So once this neurotransmitter, I mean acetylcholine, once it binds to the receptor and then the signals initiated, then it's come rapidly the acetylcholine esterase and breaking down the acetylcholine, which is active, into inactive metabolite acetate and choline, and that is lead to destruction of acetylcholine, and that prevents continuous stimulation of the postsynaptic cell, thereby terminating the nerve signal and allowing the system to reset for the next signal. The so the first function is termination of the nerve signal transmission. The acetylcholine is to play like a break for the acetylcholine. It's a break for the muscle, a break for the neuron, not continuous stimulation, just a break between the signal and the other signal. So the action of acetylcholine esterase will be so rapid. Prevention of continuous stimulation. It's the same mechanism, but just explain to us why it causes termination of the nerve signal transmission and the prevention of continuous stimulation. Just imagine if acetylcholine were allowed to persist in the synaptic cleft, or just imagine there is nothing called acetylcholine esterase. it would cause prolonged stimulation of the postsynaptic cell, continuous muscle contraction, abnormal signaling in the nervous system, and that is not good for our system. That is not normal, you know. So if acetylcholine were allowed to persist in the synaptic cleft, it will cause prolonged stimulation of the postsynaptic cell, which just means continuous muscle contraction, abnormal signaling in the nervous system. But by breaking down of the acetylcholine esterase prevent overstimulation of the postsynaptic cells, which ensuring that the neural signal is brief and precise. That's what we want. That is the normal and the physiological, that the neural signal is brief and precise. Recycling of choline. Maybe the question is, what is, the fate, we can say, of the acetate and the choline? After acetylcholine is broken down by choline esterase or acetylcholine esterase, choline is transported back into the presynaptic neuron, where it can be reused to synthesize a new acetylcholine. So there is no waste product. Choline, it will be again recycled in the presynaptic neuron to be reused to new acetylcholine. The recycling process is essential for maintaining efficient neurotransmission, especially in the regions of the body with frequent neural signalings like muscles. So this is for the first question. Just we see how that is can be happen. This is the presynaptic neuron, and this is acetyl coenzyme A with the choline, and then it lead to formation of acetylcholine, and then the acetylcholine bind to the receptor that is present in the postsynaptic neuron, and this is the synaptic cleft. Is come rapidly acetylcholine esterase and lead to breakdown of the acetylcholine into acetate and choline, and then choline recycled back to the presynaptic to resynthesize again to form acetylcholine, which is normal, is like a brief and short neuron signal, so we prevent the overstimulations of contraction or something in the postsynaptic cell. The second question is, how many classes of G protein coupled receptors, we call this like, or we can say G proteins, do you know? So we have we have 5 classes. The first one is a class A G protein coupled receptor, and it's known as rhodopsin like receptors. And this is a huge number of the receptors. So the first class is called the class A GPCR rhodopsin like receptors. The number of the receptor is 701, and the examples like muscarinic acetylcholine receptors, adrenoreceptors, histaminergic receptors, serotonin, they all like rhodopsin like receptors. So the class A is called rhodopsin like receptors, and it contains the largest numbers of members. It's a 701. Muscarinic receptors, adrenoreceptors, serotonin, histaminergic receptors. So we have large numbers of receptors. The other class is called class P. So we don't have it like A, B, C, no D, F. Because P, we have 2P. We have a class P1 receptors, and we have a class P. So about the class P1 receptor is called secretin family. From its name, secretin family, it's about the secretion, and the number of the members is 15. Okay, examples like glycogen like peptide 1 receptor, and we have a growth hormone releasing hormone. So these are the examples of secreting family. The number is 15. Examples like glucagon like peptide 1 receptors, growth hormone releasing hormone. And we have a class P GPCR receptor adhesion family, and the number of the family is 24 members. And like AD it's from adhesion, GR receptors, G receptors, lipoproteins 1, and we have adhesion G receptor GR1. Okay, so we have a number of receptors and they 24 members. We have a class C GPCR receptors, from metabolic tropic glutamate receptors, or we can say glutamate receptors, and the number is 15, like secretin, and like GABA B receptors, or orphan GPRCs 5. And we have finally class F GPCR. We called it as a frizzled or smoothened receptors, and the number is 24. It's like the adhesion family. Examples like smoothened receptor SMO, or frizzled group A, frizzled group B. So we have a number of it. So we can say the class A GPCR, which is known as a rhodopsin like receptors, the huge number, the largest number is 701, while class B GPCR, class F GPCR, it contains class B GPCR adhesion family 24 members, and also class F 24 members, class P1 secretin family, class C glutamate receptors 15. um, you can see how we can just memorize these classes of GPCR. This is the questions number 2. So questions number 3, name the levels of GPCR signaling in the right order, like what is happened. Okay, so the steps. So the first step is agonist binding. An agonist, a single-link molecule, bind to the extracellular domain of the GPCR. So agonist bind to the GPCR, bind to the receptors. Then the receptor activation. The binding induces a conformational change in the GPCR, and that leads to the activation. Maybe the question is why, when the agonist bind to the extracellular domain of GPCR, that is lead to the activation, because it's first conformational change in this receptor, and that is lead to the activation. Okay, after its activation, then the G protein coupling. The activated GPCR interact with the heterotrimeric G protein, which is consist of alpha, beta, and gamma subunits on the intracellular sites. So we say first that the agonist binding to the extracellular domain of the GPCR, and then that is lead to the conformational changes on the GPCR, that is lead to the activated GPCR interact with the hetero a trimeric, and from its name a trimeric, that is contains of 3 subunits on the intracellular side. Now the activation is happen on the intracellular side, and that is a good point. When GDP GTP exchange, the interaction, that interaction promote the exchange of GDP inactive to GTP active on the gamma alpha subunit, activating the G protein. So when this exchange happened from GDP to the GTP on the alpha subunit, that is leading to activation of the G protein. Then what is happened? G protein subunit dissociation. Then the G alpha subunit now bound to the GTP, dissociated from the G beta gamma dimer, and both subunits can interact with the downstream effectors. Like, so the first activation happened on the alpha subunit, and then when this activation, once the G or the alpha subunit of the G protein bind to the GTP, then dissociated from gamma and beta subunit, and then happens effector interaction, like the gamma alpha subunit or gamma beta gamma subunit activate or inhibit various effector proteins. Maybe what is the action of there, whatever, maybe activation or inhibition, but that lead to adenyl cyclase phospholipase C. And then a second messenger production, effector activation, whatever the function here, maybe activate or inhibit the effector. Then the effector activation lead to the generation of second messengers like CMP, IP3, DAG, DAG acylglyceride, that further propagate the signaling, and that is lead to the downstream signaling cascade. Like these events happens so quickly, and then the second messengers activate intracellular signaling pathways, often involving kinases like a protein kinase A, protein kinase C in others molecules, and then it's happened like a downstream signaling cascade. Like so much events happened so quickly. So in summary, I can say um, then the signal termination, the gamma alpha subunit hydrolyzes GTP to the GTP to the inactive site, and that facilitated by its intrinsic GT-base activity, because it contains something is called the GT-base activity, or there is regularity proteins, and then it changes GTP to the GTP, and that is lead to the reassociation of alpha beta gamma subunits and resysting of the system, and then coming back to the together. In summary, the first one is happened agonist binding, and then receptor activation, G protein coupling, GDP GTP exchange, and that is lead to the dissociation. This lead to the effector interaction, and then lead activation of secondary messenger production, and that is lead to the downstream signaling, and finally signal termination. And these are the steps of activation G protein till the inactivation of the G protein. Now I'm talking about the questions. Number 4, what types of trimeric G proteins can be distinguished because of their alpha subunit? Trimeric G protein can be classified based on the type of their alpha subunit. So we can make as a classification of the G protein depends on the alpha subunit, as different alpha subunits determine the specific, because you can see that the activation happened with the first with the alpha subunit. So the specific downstream signaling pathways and affects the major classes of trimeric G protein distinguished by their alpha subunits, and include gamma alpha S. S is the first letter from stimulatory, and the stimulatory that's mean activation. Gamma alpha I, and that's mean inhibitory, and that is lead to the inhibition. Gamma alpha Q11 and gamma alpha 12, 13, and now we want to know why they are called like this. So the first one is gamma alpha. They are distinguished, like these questions is maybe tells you by other side that the gamma and the beta subunits they are the same, but the differences in the G protein because of alpha subunit. So we have subtypes of the alpha subunit of the G protein. So we have the first one is a G alpha stimulatory, which is known as S, and that is lead to activations of adenyl cyclase, and that is leading to an increase the production of cyclic AMP, which acts as a second messenger to activate protein kinase A and other targets. So the stimulatory is activation of adenyl cyclase, and that is lead to the activation of CAMP, which activate the second messenger, and the second messenger, no, it's like they are as actually the second messenger, and that is lead to activate the protein kinase. And these examples like receptors for hormone like epinephrine and the glycogen. Epinephrine we know like adrenaline, and the glycogen it's for increasing the blood sugar in the blood. And we have also the gamma alpha I, which is inhibitory. It's completely against the gamma alpha stimulatory. It's inhibit the adenyl cyclase, and that is lead to decrease in the CAMP levels, and also regulate certain ion channels, and such these receptors, for example alpha 2 adrenergic receptors. So the alpha 2 adrenergic receptors act as a or contains gamma alpha inhibitory. We have also gamma alpha Q11 activates phospholipase C, which leading to production of inositol triphosphate and diacyl glycerol. And the inositol triphosphate increases the intracellular calcium levels, while the diacylglycerol activate the protein kinase C. So, like a muscarinic acetylcholine receptors, we have M1, M3, and M5. So, about the gamma AQ11, I don't know actually why they called it as a Q and 11, and that maybe that's we have to read the history of the discovering of such this protein. But the gamma or G alpha Q11, it's activate the phospholipase C, and the activation of phospholipase C that is lead to activation of two, we can say, second messengers like installed triphosphate and diacylglycerol. Installed triphosphate increases the calcium inside the cell, and the diacylglycerol increase or activate the protein kinase C. And the example for the G alpha Q11 that is present in a muscarinic acetylcholine receptors like M1, M3, and M5. We have also the gamma alpha 1213, and they are activation of a guanine nucleotide exchange factors, and we will discuss what they this is the second questions actually, which in turn regulate the RHO family of small GT bases involved in cytoskeletal organization, cell migration, and other receptors. And these receptors that is present in that is participate in regulating still growth or morphogenesis. So these receptors that is present in something that is related to the cell like growth and something that is related to the morphogenesis. Each type of alpha subunit lead to distinct signaling pathway, enabling diverse cellular responses to the external stimuli. So that is make the diversity of the G protein. So we have the G alpha S, we have the G alpha I, we have the G alpha Q11, we have the G alpha 1213, and now we know examples of each type. Now we go maybe the last questions. What is the role of a guanine nucleotide exchange factor? A guanine nucleotide exchange factor plays a crucial role in recoagulation of a G protein. How that is happened by facilitating the exchange of GTP guanosine diphosphate to 4-GTP guanosine triphosphate on the G protein, like recoagulation, the exchange. And we know when it's exchange from GTP to the GTP, that's mean the activation of a guanine nucleotide exchange factor. What are the key rules of the guanine exchange, or we can say guanine nucleotide exchange factor? The first one is activation of a G protein. The G protein, both heterotrimeric G protein and the small GT bases like RAS, and this is an important question, so I will discuss it later. G protein on its different forms like heterotrimeric G protein, and we said the trimeric, that's mean 3 subunits alpha, beta, and gamma, and the small GT bases which is hydrolyzes the GTP to the GTP because it's like enzyme, are usually inactive when bound to the GTP. If these G protein or GTPs, when they bound to the GTP, they are inactive. Then the guanine exchange factor promote the release of GTP from GTP, allowing a GTP molecule to bind in its place. Like the GTP is bound to these proteins, like G protein and the small GTPs, and once it's bound to these proteins, it makes it inactive. Then the G guanine exchange factor just said to the GTP, please go away, and let the GTP in your place. And then once the GTP in its place, then that is lead to the activation of G protein. Since the GTP is more abundant in the cells than the GTP, and that's a good thing, this exchange results in activation of the G protein. Signal transduction. Once bound to the GTP, the G protein adopts an active conformation. Like, it happens like, it exchange, and that is lead to the activation, enabling it to interact with and regulate the downstream signaling effectors. For example, in the GPCR signaling, the active GPCR acts as a guanine exchange factor for the associated trimeric G protein by catalyzing GDP GTP exchange on the G alpha subunit and thereby activating it. And then the final step is regulation of the cellular processes by controlling the activation state of the G proteins. G exchange factors play essential roles in many cellular processes, including the cell growth, differentiation, cytoskeletal dynamics, physical trafficking, cell signaling. So it's very important in many things. So we can say, as a summary, its guanine exchange factors act as a molecular switches that promote the activation of G protein by triggering the exchange of the GTP for GTP, thereby enabling the G protein to transmit cellular signaling. And this regularity mechanism is essential for proper cellular function and response to the extracellular stimuli. And by these questions, we end the first lectures of signaling. Thank you so much.